familiar faces from this morning, but just a quick recap. Work for a company called Treehouse Biotech, where we're very interested in isolating a lot of the minor cannabinoids that are very rare. So by our count, there's 113 individual cannabinoids to be isolated. And then my group is really just kind of trying to get those isolated so we can look at them in pure forms in cell lines, biological systems, figure out, unravel the whole puzzle, what's going on here. So today, in lieu of a title slide, I'm just going to show you what we're going to talk about. So first, I wanted to do just a quick overview of sort of what I call chemical ecology. So nature makes compounds in general, all over for lots of different reasons. So a lot of times these are useful to people, and society finds them valuable, and sort of why that is. Then diving more into what we care about, the cannabis plant. Why does the plant make THC, CBD, other cannabinoids? What's the value there? And then sort of things I see coming down the line in the next 5 to 10 years that could really change things and make things interesting. So without further ado, nature makes compounds. And so one example that we'll see in a minute here is a bombardier beetle, and that's a self-defense mechanism that's really kind of mind-blowing. On the other hand, why would nature make compounds besides a beetle trying to defend itself from being eaten? Plants can't run away. So if they make some sort of poison or something really bitter, so a deer comes by, bites a leaf, spits it out, doesn't eat the plant again. That's sort of their only mode of defense. On the other hand, you have bacteria in the soil, and they're constantly in a battle with fungi, protozoa, single-cell organisms. They don't really punch each other when they have disputes. They sort of will emit an antibiotic and kill off whatever's trying to take over their space and their resources. Even people make tons of compounds all through your body, do all kinds of different things. A good example is melanin in your skin, protects you from UV radiation. Something I won't really talk about, but is interesting as well, is a flower will mimic a bee pheromone to attract the bee over. The bee comes over. If it's lucky, the flower gives it nectar as a reward, since it got it all excited. And the bee grabs some pollen and flies away and pollinates other flowers. And biofilms have been sort of a big thing in the medical community lately, where you see bacteria start to send out little chemical signals to say, let's form a biofilm together. So just a few examples. So the bombardier beetle is like the craziest animal ever. It has 2 sacs at its stomach. One holds a compound called hydroquinone, and one holds hydrogen peroxide in these huge reservoirs. And when it's threatened, it mixes them together. They undergo a chemical reaction, gets really hot, and it sprays out this 100-degree white spray foamy thing, which blinds you and makes it hard to breathe, and is really, really unpleasant. And it also has this nozzle here that can turn 270 degrees and spray in a really wide radius. So it has no natural predators, as you can imagine. So that's just one example of when nature makes compounds to protect itself. Very common one. I grew up on the East Coast, poison ivy, big thing. You touch it once, you know it, and it's just a cute, terrible, terrible thing. You stay away from the plant. I love that. That's great. So you can imagine a deer walks by, takes a bite of the plant, has a really terrible day, never eats the plant again. It's sort of a self-defense. Talking about microbes in the soil, penicillin is the first big example that comes to mind. So by accident, this mean-looking guy here, Alexander Fleming, discovered penicillin. So he was trying to grow bacteria, and he did a really terrible job and got some fungus on the plate. And in the morning, he saw that where the fungus was growing, no bacteria were growing in this blue area. And so this had been seen before, and people just figured, bad plate, threw it out. And he actually looked and found that the fungus was making penicillin. And then he was a really terrible speaker and orator and writer. If you can't tell from the picture, he just didn't like people. And so he didn't communicate this idea very well. And so it was almost 20 years before people started to catch on to what penicillin could do. And then you see it over the counter. Just walk up to your pharmacy, say, I want penicillin. And they're advertising for it and all this. But that brought in an age of antibiotics, and we found 60 to 70% of our antibiotics come from soil bacteria. So this Fight Club's been pretty useful to people in general. And this is one of my favorites. Quinine powder is something you find in the bark of this South American tree. And it's used to treat malaria very well before it was overused. And so when British soldiers were trying to take over Africa a long time ago, there was a big compliance problem taking their quinine pills. And so the gin and tonic was invented because it's much easier to say, did you drink your gin and tonic today? And if you've ever, then did you take your malaria pills? If you've ever put a gin and tonic up to like a black light, if you're out at night, you might notice a blue glow, which we can all check out tonight, I guess. And so that is actually the quinine in the tonic water, absorbing that light and emitting blue. And so there's your daily gin and tonic for keep you healthy. So the fact is, plants and bacteria produce compounds that society finds useful, and then sort of why. And there's a lot of theories about that. Why is it useful to us? Sometimes it's by accident, and sometimes the plant's doing it on purpose. So in the case of nicotine, our goal is kind of aligned, whereas nicotine is an amazing insecticide. And so it really protects the plant from being eaten by insects. Native Americans found out if you're smoking tobacco, the mosquitoes leave you alone. So they just kept doing it, growing the plant, and it turned out they really enjoyed it quite a bit as well, which was probably an accident. But for a long time, it didn't. I mean, 1690, people figured out we can extract the plant and spray it as an insecticide on other crops. So that was pretty influential. Caffeine is another example that comes to mind. It's very toxic to most animals. You see birds, dogs, cats, insect spiders. And people really very, very low toxicity, and we can't seem to get enough of it. So there'd be an accident where it was supposed to keep us away, and we ended up, just getting great value from it. And then mushrooms, this is probably on purpose. If you're walking around, you're hungry, you eat some of these, then you keep walking, you could have a pretty bad day, and then you might think twice about eating the mushroom again. So that's sort of the quick run-through of what I call chemical ecology. Let's kind of move on to the plant we care about. So I'm a synthetic chemist by training, and so I promise this will be the last hardcore chemistry part of the talk, but I had to throw it in. So the cannabis plant does something pretty normal that most plants do. It makes terpenes. And so that's all done just by combining these two compounds to get to what will eventually become geraniol or some other terpene. This is sort of a progenitor molecule here. It also does something else very common. It makes a class of molecules called polyketides, which are found all throughout nature. And that's done by combining these fatty acids with malonyl-CoA, and then in a series, and you get these long chains. And in the case of cannabis, you get olivotolic acid. The unique thing that's very rare that cannabis does is it combines a terpene and a polyketide and makes cannabinoids. So you take your geranyl phosphate from above and your olivotolic acid from here, and you get CBGa, which is the progenitor of all cannabinoids. And as far as we know, this is the only plant that really makes cannabinoids with this long chain right here. And that's what gives us all the special properties we see from hemp-derived cannabinoids. So as we said, CBGa is the progenitor to all cannabinoids. So 3 examples. There are 3 enzymes the plant produces: CBDA synthase, THCA synthase, and CBCA synthase, which are all really well named because CBDA synthase synthesizes CBD, and so on. And so this is where we get pretty much a lot of the cannabinoid diversity we see. CBG, and then enzymes that the plant produces to make specific cannabinoids. Then there are other things that arise not on purpose by the plant. So if you leave CBCA out in light long enough, it will slowly convert to this really funky looking thing, CBLA. Or THC can degrade to CBN. And so we start to get this huge richness in compounds out of that. Also in decarboxylation, if you leave them out long enough, you start to lose that. This group here, which is an acid group, to get to just standard cannabinoid compounds. And so this happens if you're smoking, you light the plant on fire, that's enough heat to do this whole process and get you to the active form, which is the decarboxylated form. And same goes THC, you light it on fire, you get some CBN, some THC. So there's a lot of theories around THC because it's been so profound, and we've known about it for a long time, and it's been well studied. So I'll just run through three real quick. One idea is that THC is really good at absorbing UV light, so much like melanin protects us in our skin, could just be protecting the plant from any kind of UV light, any radiation that's bad, causes problems, and by accident, we get a psychoactive effect from it. Number two, it has been shown to be antibacterial in some cell studies. And so it could be protecting itself, from a foreign invader, and by accident, we get a psychoactive effect. Or it could be psychoactive on purpose, right? You eat the plant, you have a terrible day, you wander off, and you decide, maybe think twice. But food for thought, we really like it, and so now we've cultivated this plant all over the world. So it actually kind of worked in the plant's favor, no matter how you slice it, because it gets to grow everywhere now. And the other cannabinoids are even trickier to think about because they're not psychoactive. So why are they being made? If THC is being made because it's psychoactive, why would it be making all these other ones? So CBG is easy. You have to make it to make THC, so it's just going to be there. But on the other hand, CBC doesn't really seem to do much for us. So it turns out it's slightly antiviral. There's a lot of promising stuff there. But they all sort of absorb light well, so maybe the protecting from UV light theory isn't so bad. So just kind of running through that, just want to talk about sort of future things that are happening now that I think we should keep our eyes out for moving forward. So there's been a lot out about transgenic stuff lately and GMOs. So you'll see transgenic tobacco is one example. So tobacco has been very well studied. The whole genome is mapped out. We know how to grow it really well. It produces a ton of biomass. And so there is tobacco that's been engineered with the THC-A synthase put into it so that it can produce THC. And it even has trichomes like you see on a cannabis plant where the THC is growing. The catch is you have to feed it the CBG. But we're one step away from just engineering in that CBG production, and then suddenly you've got a tobacco plant making THC. Also, you guys might have heard of the yeast that's been modified to make cannabinoids now. And so, I think that's where a lot of things are headed, because you grow that up just in a big reactor rather than this huge field of plants. So it's a lot smaller. Your input is sugar instead of all the farming time and labor and water and nutrients that go into that. I will say it's not a great yield yet. There's a lot of room for improvement. It needs to be optimized. We're not talking about really a reactor yet. We're talking about sort of this scale right now. So it's not really enough to disrupt any kind of farming right now or really make a big dent. And this has been done quite a bit with other compounds, morphine, for example. Some great work done at Stanford, and it's been translated, but it hasn't really hit that big scaled up point yet. Something to keep an eye out for. There's also CRISPR, which is a little different. Rather than adding a gene in, you're taking a gene out. So this is pretty new technology. CRISPRs are sort of self-defense mechanisms that bacteria use to cut out viral DNA when a virus invades them. And it's been engineered by people and now can be used to target specific genes in any organism. And so you're literally just pulling out a gene of interest rather than adding something in. And so these arrays were identified in the 80s, but it took a very long time to figure out exactly what their function was and what they were doing, about 2005, before we actually knew what was going on. But then you see in 2013, the first 5 papers of a CRISPR-modified plant coming out. By the end of 2014, 1,000. So it really took off and got huge. And then in 2016, mushrooms came out. And this is where the mushrooms usually brown when they get old, and then they're on the line in the next 5 to 10 years based on the explosion of research coming out and these mushrooms coming out, all this going on. Something to keep an eye on for sure. So that's sort of my whirlwind through all the science. That's kind of where we're at here. So just in summary, nature makes a huge diversity of compounds to use for all kinds of different reasons. We find them very useful much of the time. The cannabis plant, specifically now, we're really starting to look at, and you have these discrete enzymatic pathways that can start to be manipulated coming down the line. So a lot of modern technology on the horizon, maybe. For better, for worse, we'll have to see.
Thank you, Dr. Robert Gray. Do we have any questions?
Just curious about CRISPR. How does that actually work that they would discover which are the genes to take out? I mean, is it just trial and error, and is that why it's going to take 5 to 10 years to do this?
No, I think okay, so how it works. So CRISPR sequences are these repetitive units that an enzyme or a protein will recognize. And when it recognizes that, it'll come down to wherever, let me back up. I should have put more in on this. This is an interesting thing. So in the bacteria, viral DNA comes in. They have a protein that adds this CRISPR sequence to that DNA. So then the virus, it inserts its DNA into the host DNA. But now you have this CRISPR sequence with it where a protein can come identify the viral sequence now, because it identifies the CRISPR, which is attached, and it just snips that out. And so what researchers have done is essentially take the sequence of, say, THCA synthase and appended a CRISPR unit to that. And so you add that to the cell, and now you have this whole host system that can just identify the CRISPR, cut out the THCA. So you can't just, there are complications with going in and just snipping things out. So I'm guessing 5 to 10 years before we see this, but with science, it's just, sometimes you get lucky and it's quick. Sometimes there's, things you don't expect. It takes a long time. But you saw in the 1 year span, quite a bit come out. And so it seems fairly universal. One thing I will say is, it hasn't been studied well enough to necessarily see, is it very selective? Is there off-site stuff going on or other things getting snipped that you might not have intended? Also, I'm of the opinion, you cut something out and it seems to do what you want, but what are the downstream consequences of that you can't predict? So I don't know. There might be some regulation coming in to address this, but right now it's relatively unencumbered.
Any other questions?
Can you elaborate a small bit on the complications that we're encountering?
Hold on. We need to get your question on the mic, you guys. We're recording.
Thank you. I was wondering if you can elaborate a bit more on the complications we're currently encountering for the 1 to 5 year process. I have the same mindset. If you have a revolution, a cycle, or something, producing THC and you're removing stuff,
Who knows?
will it affect it eventually? So it does take a process, right?
So the plant makes it for a reason, right? So you take it away. Maybe you don't know that reason, and you figure it out, hindsight after the fact. So penicillin's a great example. It was sold over the counter. People were taking it when they had a cough. It's everywhere, and then you get antibiotic resistance springing up, and nobody saw that coming. And now it's a huge problem. And so with the CRISPR, I just more say, you don't know until you do it. And that's more my concern is the things you can't guess. Who knows? It could make the plant less drought resistant. You have no idea. You have no idea why it's making the THC in the first place. You only have